Welcome to lecture 10. So today we're going to study methods in microbial ecology and microbial habitat. And so we'll first look at the methods, and then we'll look at the several different ecosystems, and then look at the application. Ultimately, if we would, one of the goals to understand microbes in their environment is hopefully we can take advantage of them, and then or avoid them, right? And so we'll first look at the methods. So the microbial ecology is actually a big field. You know, ecology is to study the organism in their own environment, ecosystem. And there are different areas. Microbial ecology is actually a big area of study, and microbes obviously have huge impact on us. Indirectly through ecology, and so if you are interested in environment, that's one area that we'll be studying. There's, if you don't know, there's actually a society interested in or has been working on restoring Bronx River. If you didn't know, Bronx does have a river called Bronx River. And it runs through Botanical Garden and the zoo, around Bronx Zoo. And that society was interested in restoring, and Dr. Racklin in our department was involved in restocking the fish. So you can go fishing in Bronx River now, thanks in part to Dr. Racklin's work. But there are people interested in monitoring bacteria, because how do you know the water is clean? Well, it's probably not clean, but how unclean, right? And so are there sewages in there? One of the ways you monitor is to monitor the bacteria. And so I was contacted by the executive director of Bronx River Restoration Society, I guess. So I looked at that, and then they actually did monitor, and they got an EPA grant to monitor the bacteria. So we actually chatted, and so I told her that we have students interested in microbiology, and we have all the equipment. Maybe we can have a collaboration with them. So after today's lecture, you'll be interested to know. So there are many ways of monitoring. Basically, is to counting the numbers of bacteria. One of the ways is culture dependent, meaning letting the bacteria grow. So the culture dependent methods involves two important concepts: enrichment and isolation. So enrichment, this is important because in nature you can have many different species of bacteria. They live together, and they're all unique in a niche, right? And they depend on each other for food. And holidays coming up, and you depend on which microbe to provide you with one of the key entertainment, like beer, wine, and that's from
yeast.
Yeah. Yeah, it's a yeast waste, right? It's the waste product of the yeast. I'm sorry. You can go about then and tell your friends. Maybe that will stop them from drinking too much. But for bacteria, we depend on yeast for entertainment. But bacteria actually depends on each other for energy consumption. They're not interested in entertainment. They're interested in reproduction, and so they need energy. And so the point is that in any environment, you have a mixture of many different bacteria. So in order to get them, you're going to have to increase the number. Okay, so the way you think about it is if you get a bucket from Bronx River of water, and then if you get 100, if you get 1 liter, right? So we go by 1 liter. If you get 1 liter of water from Bronx River, it has many different bacteria, and the bacteria that you're interested, maybe you have 100 bacteria per liter, right? And so let me just start typing in, maybe this will. So if you got 1 liter, that's 1,000 ml, right? And and then the bacteria, and then you have 100 of your bacteria of your interests, let's say E. coli, right? And so now, if you take 1 ml, and how many E. coli per 1 ml. So if you take 1 ml of that Bronx River water, how many E. coli you will get if you take just 1 ml? No one? You won't get, if it's strictly speaking, the chances of you getting 1 bacteria in that 1 ml is 1 in 10, right? And so in theory, it's 0.1 E. coli, but that's not possible, right? So what you then do is you provide. So what you do is add nutrient, right? And grow overnight. Now, if you let this 1 liter of bacteria grow overnight, and that 100 E. coli becomes 100 right? At least, okay, at minimum. So now after this, how many E. coli per 1 ml? Oh shoot. Okay, sorry about that. Can you see now? That's why you guys are quiet. About that.
Yeah, I can see it now. I was waiting for you to send it.
We can see.
Oh no, because I made a mistake, because until I finished typing, you wouldn't see it. But now, okay. So let's go back. And so if you take 1 liter from Bronx River, and assuming it has 100 E. coli cells, and so the question then is how many E. coli per ml? If you get 1 ml, chances are you won't get any E. coli, right? And 1 in 10. Not 9 out of 10 times that 1 ml doesn't contain E. coli, right? And so now, and then, but if you add nutrient to that one liter and you let it grow overnight, that 100 E. coli becomes 100 million. So then the question is, okay, hold on 1 second. So now the question is, how many E. coli per ml now? So that becomes 100. That's 100 million, right?
Yeah, so would it be 1 million now?
No. It'd be 100. No, it'd be 100. Right? 100,000.
Yeah, if you just move the decimal point over a couple places, it's 100,000.
Right. So that would be. So now you see the problem is now if you just get the river water from Bronx River and you say, God, it's clean. It has no E. coli, right? Because you sampled 1 ml and didn't find E. coli and say, great, it's clean. But then you say, well, what if it blows my detection level? It's there, but I'm not getting it, right? So now you can say, okay, I add nutrient and let it grow overnight, and then E. coli will turn out to be this much. And so now when you get E. coli, you will get it, right? So this step here, this is enrichment. Okay. This is the concept enrichment. You let it grow, and with this though, depends on the nutrient. If you add a nutrient that allows E. coli to grow, but when other bacteria also grow, then that's considered to be that's not good enough, right? Because you increase E. coli, unless you can you can see the selecting the E. coli, then you can see it. Okay, so let me make it more stringent. Let's go to the next slide. So now hopefully you have a little bit knowledge. So now if you look at this particular slide, what they want to see is, the question is counting the is there nitrogen fixing bacteria? So what if you want to isolate nitrogen fixing bacteria? Now the problem is slightly different, right? So the previous problem is counting E. coli, and we simply threw a nutrient and then count all the bacteria. That's actually it's not. And so now it's you start from the solution, and with the so if you need to be careful with reading here. So you put together a nutrient solution with salt and mannitol. Mannitol is a sugar to provide carbon source, but you do not provide nitrogen, either inorganic or organic form, right? So in this bottle, and then in this you put soil. What's in the soil? Soil contains lots of bacteria. And then you put the soil in there, And then after that, what you do, you pipe in, you use a pipe to provide the nitrogen gas and oxygen gas, and you bubble it. So in this condition, what bacteria will be able to grow? You did not provide the nitrogen. So and we know bacteria and all living organism requires nitrogen, right? Because amino acid. If you don't have a nitrogen, fix the nitrogen, then you cannot produce amino acid. And if you don't have amino acid, well, you can't make proteins. You can't do translations, no enzyme, and you can't grow, right? So in this condition, the only bacteria that can grow is the bacteria can overcome the nitrogen problem, right? And then there are bacteria that can turn atmosphere nitrogen into ammonia, and this is the bacteria called nitrogen fixing bacteria. Remember carbon fixation? That's Calvin cycle, right? And carbon fixation is turning CO2 into glucose eventually, right? And so here you have this nitrogen fixing bacteria. Now, what about the other bacteria? So if you have E. coli, let's say the soil has E. coli. So if you have E. coli in here, will E. coli be able to grow? E. coli doesn't have the ability to fix nitrogen. If you don't know, that's the condition. So E. coli cannot fix nitrogen, so it cannot grow. So the only bacteria that can fix nitrogen is Azotobacter. Remember how expensive is to fix CO2? Remember, we calculated, right? To produce one glucose molecule, you need at minimum 54 ATPs, right? Now, nitrogen is a very, very, very stable molecule, has 3 bonds between the 2 nitrogen elements. Okay, so it's very stable. And so it requires huge amount of energy to break them up. Okay, so you need a much stable, you need lots of energy, but in the soil, the energy source is very minimal, and so in regular soil, and so most bacteria do not fix nitrogen because it's too energy demanding. Okay, so that's one condition. So you have this, and then we can duplicate this. Oops, what did I do? Sorry. I'm just going to type it here. And okay. So now, if you you can change the condition. So the same media, okay. So it's if you change the media composition, now if you add ammonia, the only difference is now you add ammonia. Okay, so these two here are different, right? So now, if you look at, so you have Azotobacter, and then you have E. coli. Let's assume these bacteria have these two, right? Now, if you look at here, and then you, assuming these two bacteria in the soil, and then you let it grow. And then after that, you can, then you take 1 ml, a small amount from the liquid, and then you put it on the plate, and you actually streak them on the plate. Okay, so you plate, and if you plate on here, because when you plate, it's actually small. Let me put a number here, and usually it's, sorry. .1 ml. Okay, so you put 0.1 ml on the plate. Now, in this case, you will see, and you put them on two different growth conditions. On this plate, you omit the nitrogen. On this plate, you provide the nitrogen. Okay, you will see that in this condition, when you change this condition, there is no bacteria here. Versus in this condition, there is a bacteria, and you still, let me just type it here. Okay. Now, so the difficult part for people to see it the first time is, well, on these two media, they're the same. In this condition, you see bacteria, and these are the azotobacteria, azotobacter, but E. coli did not grow. But in this case, what happened here is that the azotobacter, did the azotobacter grow? No, it did not, right? And on this plate, in this growth media, which bacteria grow? It is the E. coli. Well, because the E. coli doesn't need, it cannot produce nitrogen. So it depends on the nitrogen source. But if you provide the nitrogen source, E. coli will grow huge amount and take up all the nitrogen, take up all the carbon source. So in this case, well, here, the carbon source was taken by E. coli, right? So now E. coli is able to grow. So you have lots of E. coli, right? So this is really the essence, is one of the best example to explain the enrichment. Different conditions, you can enrich, you can create condition that you favor one particular bacteria, and that is enrichment. Okay, so once you enriched and the numbers of bacteria increased, because you increase in this enrichment, now in this condition, lots of E. coli. And right, because azotobacter, let's see, because azotobacter don't carbon source. because azotobacter lost in the competition for nitrogen source, right? Because E. coli took all of them. Okay, so all right, and so I'm going to pause here to see if everyone is still with me. Questions? Okay, so let me rephrase this particular challenge here. So the concept here, the enrichment concept, enrichment is you create a condition favor one bacteria grow. Okay, so if you want to isolate a nitrogen fixing bacteria, you will create a condition that only the nitrogen fixing bacteria will be able to grow, the other bacteria will not. Okay, so in this case, if you want to isolate a nitrogen fixing bacteria, then what you do is you put a growth nutrient with lots of carbon source, but with no fixed nitrogen. And the only bacteria then can grow in here is the azotobacter. Okay, so here in this condition, you have lots of azoto. Oh, does it show up? Yeah. And you have lots of azotobacter. Okay. Lots of azotobacter in here. Why? Well, because that's the only bacteria that can grow. It grows so much that if you just take 0.1 ml out, you can still see it. Okay, they're all there, but azotobacter become the dominant species. And in contrast, I'll give you a control. Biologists always need to do a control. As a control, what you see is, what if you do things differently? So what if you give nitrogen source? If you give nitrogen source, then bacteria like E. coli, any bacteria, majority of bacteria, if we think about this too, using E. coli example, so E. coli in this condition will grow, will take up all the nitrogen, the nitrogen bacteria, the common source, and then become the dominant bacteria. So E. coli becomes the dominant bacteria in this case. All right. And then it becomes so dominant, you just need 0.1 ml, and then you can detect the presence. Okay. So the question, let me go to. Okay. So Melody asked, there was a selective agar. So the selective enrichment is different from selective. Okay. So enrichment, where do I find? What do I say? Let me put it on here. Enrich is favor one bacterium, and selective media to grow. And kill all others. Okay, so to answer Melody's excellent question, there's a difference. The difference between enrichment versus selective is the enrichment you provide the favor conditions for one bacteria. That's your favorite. But you don't care about the others. You don't kill E. coli in this case. However, for selective media, and what you do is then you create a condition that only allow one bacteria to grow. So for example, if you know there's an antibiotic, if you know you have a bacteria that can resist, say, a particular antibiotic, then you include the antibiotics, that particular antibiotics, in the media. And so then the only bacteria that you get, or the only bacteria that can grow, is the antibiotic resistant bacteria. Okay? It's good, Melody? I'm going to return to file. Great, thank you. Yeah, next time don't, but if you have a question, just say it, because then it's because now I'm gonna have to go back to the slides. It'll be difficult. Okay, so the next thing I wanted to say to discuss is the other ways. Are there any other ways to enrich? So I'm returning to enrichment. Okay, it's culture dependent, and so another way is that there's a setup called Winografsky column. Winografsky, you saw there was a nitrogen fixing bacteria that earlier we talked about, and he's a Russian early Russian microbiologist, and he did a lot of analysis. And Winografsky column is he made famous. It was very simple. He figured out he just gets some mud and put some lake water and put it in a glass column. Literally, it's just like a glass cylinder. And then just put it under the sun at his window. And over time, and you notice it's a foil cap. It's not sealed, okay? It's very important that you notice it's a foil cap. It's not completely sealed. It's not a stopper. Why? Well, because we need oxygen to go in. Now, because the water in the column, it's not circulating, right? Because the water in the river circulates, and the water churns. The oxygen is uniform. But here, the water sits there still. So the oxygen then has high concentration at the top, low concentration at the bottom. And then you have hydrogen sulfide being produced at the bottom. Okay, and and then you have the hydrogen sulfide concentration highest at the bottom of the mud and decrease. So just for these two conditions, and then because you exposed to the sun, the energy, the ultimate energy will all come from the sun, right? Because this is a sealed system. The only thing that you input into the system is the energy. With the energy and the oxygen and the nitrogen in the air, what will happen is that some bacteria will be able to grow. So if you have one bacteria, purple sulfur bacteria or purple green sulfur bacteria, they will be able to grow in their own favorite conditions. And because the oxygen and the hydrogen sulfide concentration is a gradient, and so then these are then be able to grow. So that you can actually literally get a patch and then get the bacteria that you want. So this is another enrichment. Another different enrichment scheme or method is by extinct dilution. Right. So if you have 100 ml, so you start with this 1 ml of liquid, and if you do 1 to 10 dilution, 1 to 10 dilution, and if you have 100, if you have 100,000 bacteria, and so you will keep being diluted, and you understand at some point you're going to run out of bacteria per ml, right? And so now so at this dilution there's no bacteria, but chances are the next one will be you will have 1 or 2 bacteria there, right? And chances are if you have 1 bacteria in here, then okay. So the way you think about this is that if you have we have 100,000 bacteria in here, right? So that's your per ml. And now after 1 dilution, what you have, right? So here you have 10,000, and then here, I need to click again. So now you have 1,000, right? And then you have 100, right? And then in this tube you have 10, right? And in this tube you have 1. And in this tube you have 0, right? So obviously if you have 0 bacteria, then you won't have it. But if you have bacteria and then forms the culture, now this culture comes from 1 bacteria, so it's actually a very pure species, very pure, and you just amplify this 1 bacteria. Now if this 1 bacteria represents 1 species, then you just isolated that 1 species from nature. Okay. Questions? Anytime with number is always somewhat confusing. Okay, so the point is that if we start with a high concentration of bacteria that's in nature, Bronx River, and then you just keep diluting and diluting and diluting until there's no bacteria, and then you go back to the next tube. The chances are this tube will be formed by 1 or 2 bacteria. Now, in reality, what's going to happen is you take this tube and then you dilute it 1 more time. You do another series, and then you will find you probably have 2 or 3 different species after the dilution. And so then you actually will be able to see how many species you have. Okay, so those are the culture dependent. There are physical methods to isolate bacteria. One of them is this. Hold on. Wait, hold on. Okay, good. So one of them is using laser. So what turned out to be very interesting is that if you put a bacteria in a glass tube, uh, very, very small, capillary size glass tube. How small? It's about the size of bacteria. And and we know E. coli is 1/3, right? And so then what happened is that you can use laser, has the property of grabbing the bacteria. So if you shine focus a laser on it, and if you move the laser beam, the bacteria can actually be moved physically. It's just amazing. And I don't really understand how it moves. Physicists told us that they can do it, and they've demonstrated. And so then if you move that over here and then you cut it here, so now you just isolated the bacteria. Right? So that's a physical method of isolating. So those are the culture-dependent method to isolate and then count the bacteria. Right? Now there are other methods to count the bacteria. One, you can count on different things. You can count total number. You can count the viable number, and you can track specific bacteria population using antibody. And if it's because antibody can bind surface molecule, and then you can know, okay, if you have antibody against a protein, particular protein, then you can count how many bacteria have that protein. Now, if that protein is unique to E. coli, then you can use this method to count the E. coli. And then you can also tag living cells to study bacteria in their own environment. So let me give you an example for each. So if you want to count the total numbers of cell count, you can count DNA. And you can use a fluorescent stain called DAPI. Basically binds to DNA molecule and show blue fluorescence. And so each blue spot represents a bacteria. And you can also using what's called a viable stain. And so the living bacteria will be green, and the dead bacteria will be red. And so usually what happens is that they use the green stain is a tiny, this viable stain consists of two stains. And so the green stain is typically a tiny molecule that can stain cell green, every cell. And then the red is the bacteria that that the molecule is big enough that it cannot get into the living bacteria. It has to wait until the bacteria die and the cell has holes on them, and then the red stain goes in. Okay, so that's how it works. And so you can tell the living cell versus dead cell. So here's an example of fluorescent antibody as a cell tag. So you can have the antibody recognize a surface DNA molecule and a surface protein, and then the antibody is linked to a fluorescent material. And so then when body binds to a bacteria, and then the bacteria becomes green in this case. All right. Now, this is an example of you trap bacteria in their own living environment. And this is, let me show you. So this, what you're looking at, red, is a root hair. And you can see the little specks of green, and those are bacteria inside a tube-like structure. Okay, and this picture was the first time that we were able to see bacteria, a nitrogen-fixing bacteria, going into plants in real time. Okay, and this picture is from my my own research when I was a postdoc at MIT, and that was my paper. And so this figure came from my paper published in JBAG. I'll come back to this at the end of the class, all right, and explain more, and you will understand. And so what we just talked about was the total cell count, viable cell count, and tracking the antibody and then tagging the bacteria. And you can also, using genetic methods, to what's called a phylogenetic stain. It's called a FISH, and it's actually fluorescent in situ hybridization. And then you can also use PCR to specify genes. So if you think about, step back, think about a microbial community, right? You have lots of different bacteria. And you can see that this is a picture I showed you earlier and say, well, look at the sewage, right? Sewage is a horrible thing. It has a horrible image. But now, from this point on, you can see sewage is beautiful, right? And so why is sewage beautiful? Well, okay, so here is what happened. The FISH probe. So let's go back to here. And so you have each bacteria. Let's say this is E. coli, right? This is E. coli, and then this E. coli bacteria has lots of chromosomes. Okay, so that's its chromosome, and then change color. Let's say E. coli is red, and then you can have a DNA that forms complementary. I'm sorry, no. Let's change color. Delete. How do you delete this one? Oops. Okay. So if you have DNA and you have let's not do that. And you have a neutral, unlabeled complementary DNA, right? So that's your probe. That's the probe. Probe DNA. Probe has two parts, and then then at the end you put a red fluorescent beam here. Okay, so now you see the probe, the FISH probe, binds to the DNA, and now it can actually this bacteria becomes red. All right. So this is what the what does it mean by fluorescence in situ hybridization using a probe. So if your probe has if your probe, this is your probe, and your probe has a fluorescent dye, and then that bacteria becomes red. So if you can try different colors, so you can have blue, you can red, green, and so that's why the sewages of different, with a mixture of different bacteria, have a whole rainbow of color. Then each color, that's the part that each color represents a particular bacteria. Good? Any questions? Another way is to using phylogenetic probe. And so what that means, if you get the community, extract the DNA, and then you PCR, you amplify all the 16th sRNA gene. Now last lecture we said the 16th sRNA, what that is, and it can be used to generate as a common materials to sort bacteria, right? We said shoestring, and so if you get all the DNA, get the 16th sRNA gene out and extract, you can either go directly sequence it, or you can separate them into different samples and then sequence them. Either way, you determine the sequence of the 16th ribosome RNA from different bacteria, because you have a mixture of them, right? Because you start with a community, you have all these different bacteria, and you extract, and then you do sequence. You determine the DNA sequence of all these 16th ribosome RNA, and then you compare them. Okay. Now, we already know, like the last lecture, we already established what bacillus is. Bacillus, different species of bacillus, and then you also have Clostridium, and so if you isolate an unknown DNA, unknown bacteria, and then you just look at how similar, which one is similar. So this unknown is most similar to Bacillus cellulus, right? You look at the percent similarity, and then this unknown is similar to Clostridium, but it's halfway. And so if you look at this unknown, and this unknown is most close to the Clostridium, right? So this is how we place them. And this one is less, but it's similar to other bacillus. So this unknown is similar, somewhere between bacillus and Clostridium. And so using this type of analysis, then you can establish the phylogeny and say, okay, get a sense of what kind of bacteria that you have in the environment. Okay, I'll pause for a little question. Okay, so other physical methods we can use to study microbial ecology one is to use microelectrons to determine the size, determine the microenvironment. So, for example, you can have electron pH probes or microelectron electrodes like a pH, and you can notice it's so tiny, right? So if you were to draw an E. coli, 1/3 would be like this, right? This is an E. coli, and that is how small. And so in theory, if you do it right, you can put this probe next to the E. coli and then watch the pH change or any other things that could change. And so with that, you can monitor if you have the right, and you can measure different things. You can measure pH or sulfate or different chemicals. So in this case, you can see you can measure sulfate reduction is coupled with hydrogen sulfide increase, and you can also measure different things. So if you measure CO2 incorporation, and you can see with the light, yeah, you can measure the CO2 in more with light. You can actually see carbon gets incorporated, right? And you can also measure the hydrogen and all these other chemicals, right? Now you can take the same approach to measure water column in the in that lake, and so for example you can measure oxygen and um, and then the pH. So the oxygen is here, and this is at pH, and so this is measuring pH 6 to 7, and then you can see the pH actually changes around. Okay, because this is measuring the hot spring mats, and so if this is the water surface, and then you have lots of growth on the surface of the water, and you can see things are different in the mat. The oxygen being consumed, so the oxygen actually decreased, but at water surface, that's where the oxygen is highest because, and then it decreases as the water goes down, as you go deeper, right? And so that's physical measurement, and there are also other interesting things that you can measure. So, for example, you can measure the different carbon, different forms of carbon, the CO2, C12 versus C13. And as it turned out, remember the Calvin cycle, right? Calvin cycle, there were two enzymes that fix CO2, and the enzyme actually, when they fix CO2, these enzymes actually have a preference. They prefer C12, okay, versus C13. And so so after the carbon fixation, the C12 ratio actually increase, so much so that you can actually see the difference. The plant material. So you can have Calvin cycle plants here. So this is a zero ratio, and you see after the carbon fixation. I'm sorry. This is the natural environment, the CO2, right? Atmosphere CO2, and from here, and you can see an increase or a decrease of C13 proportionally. You see, go back here. So after the fixation, the C13 percentage decrease, well, because C12 percentage increased. All right. So so that's the plan, and then the range in the petroleum actually increase because there's other bacteria in the methane. It's even higher. The C13 concentration is actually lower. So this gives you this is really just a fun fact, just to allow you to see the living organism actually have a preference, so you can track them. And so in this case, human, where we are, if you look at the carbon ratio, what is our C13 percentage will most look like? If you were to place human on this graph, where would you put the human? Anyone want to give it a try?
Would it be with methane?
No, well, we don't eat methane, right? We eat. Do you have salad? Oh, I'm sure we all eat salad. So we mostly eat the plant material, right? The food, most of food comes from plant, and even if when we eat meat, our meat, chicken, and all those, they eat plant material, right? And so majority of the carbon that in our body should be very similar to carbon cycle plants. Okay, so that and so those are the physical methods, and I know it's could be difficult to follow. So I hope you will review and have questions. So now I like to go to ecosystem to give especially focus on a few systems. So when you look at the an ecosystem, there are a few things in general we need to talk about. Is the concept of population, community, and guild. And the population just is a general term and describes all microbes. But then you can have community, and then you have guilds. A community, so here gives you an example. A community is a physical area, and you can have a community. Does a photonic zone, does oxygenic phototrophs are living here because they're closer to the surface water, and they do photosynthesis. And then you can have anaerobes or facultative, because they may be able to ferment. And then you can have a community at the in the mud. And within each community, you can have guilds, and those are loose association, but somehow they have similar metabolic features. Okay, so you see here example they give you is methanogenic bacteria, the CO2 to methane, or homoacetogenic bacteria. They go from CO2 to acetate. So what's common is they all use CO2 as a carbon source, right? So that we put them together in a guild. So these are some of the examples. Now, the diversity in the soil is tremendous, or in nature is tremendous. Here, we're looking at, if you were to look at a soil particle, a particle of soil, and then you measure the oxygen level. And this is what you will find. The oxygen in the middle of the soil particle is 0. And as you go out and go to the very outside is 21. Why is 21? Can it be higher? Oh, is it too easy? It's 21%. This is percent oxygen, right? So 21 is what's the concentration in the air, right? So and here's an example. And so if you put a glass slides into soil, and then you come back and after a few days, and then you actually can take a look and see what is going on the soil, right? And then if you put a glass into Bronx River, and then what will happen is that there will bacteria grow on the glass, or similarly in the soil, but the bacteria come around and then they will attach, and then they actually, because the river will flow and with all the nutrients and the bacteria actually grow and produce polysaccharide and produce a film on the surface. And so this is what biofilm, okay? Biofilm refers to this part, and this is biofilm. And here is a real example of lab-grown biofilm. If you do a cross-section, and you can stain the bacteria on the surface. And so this is a cross-section of biofilm that you can actually see. Now, and so if we were to now focus on soil and fresh water a little bit more, then you will see the soil. So if you have a soil column, and this will be, if you take, a cross section of the soil, like just outside in the somewhere in Lehman College, and there will like grass grow on the surface, and then if you go deeper and deeper and deeper, you can cut them, you can group it into different different areas. And the first area on the top is where the most bacteria grow. Why? Most bacteria will be here, right? Is because the plant gets the energy from sun, they will grow, and they will secrete their waste product into the soil, and most of their waste product then being used as energy and nitrogen source, energy and nutrient for the bacteria. And so this, wherever there's a plant growth, that soil is most rich for microbe. And then the nutrient can, once the nutrient gets absorbed and some gets filtered back down and along with the water. And so the deeper you go, the less nutrient there is. All right, so that's an important concept. And so this is a better look at a soil particle. A soil particle can trap air here, and now there's air and there's water, right? So within a soil particle, aggregated soil particle, you have many different environment, and so different bacteria can grow. Okay. So here is some examples of bacteria on the surface of bacteria, different, they can grow and they get nutrient, they'll form. Now, it's interesting is that even if you go to 1,500 meters down into the earth, you will still have microbes, right? Because some of the nutrient will eventually leak all the way to the bottom, to that depth. And then they have different bacteria be able to grow. And so some will be able to generate methane, so use hydrogen, and those are the, and hydrogen production is proposed in organic. So where the hydrogen comes from? Well, the ion, oxidative ion, meets water and then releases and gets oxidized, and then hydrogen gets released, and that provides the energy source for these microbes, right? And if we were to look at the water environment, and so this is a water environment. This looks like a lake, and the water is standing still. And so if you look at this water, so what you will find is the oxygen in the air is 21% stable, the temperature is stable. But when it gets to 5 meters down, the water is still on top, microcirculates, but when it gets to deep, there's always a thermal layer. That water gets so cold it doesn't move anymore. And because the water doesn't move, the oxygen level goes down quickly. And in the water below the thermal layer, the oxygen is very low, and the temperature obviously very low. So if you go upstate New York and see a beautiful lake, don't jump too deep into it, okay? Because you're gonna hit the thermal layer, your muscles are gonna crank, and depends on your swimming skill, you may get in trouble. And then the hydrogen sulfide concentration will actually get increased. Okay, so this is tracking the hydrogen sulfide concentration that's increased. Now, if you have a river, what's going to happen is that the river will have high concentration of oxygen, but when Bronx River runs into a sewage treatment plant and the sewage comes out into the river, and well, lots of bacteria gets released, organic carbon, and so that will increase. With that increase, then you have the nitrogen, right? All these nitrogen source increase, and because oxygen, because all these bacteria, lots of oxygen is being used. So then you see a decrease of oxygen concentration. Okay, now, and the algae or cyanobacteria, they are there. But because when you have bacteria outcompeting with all the energy source, the cyanobacteria and all these, the growth of these bacteria actually decrease. But as you go away, because they have these bacterial growth actually breaks down a lot of small nutrients, and then these unused nutrients being used by algae and cyanobacteria, and those will have a boom increase. But then as you go further downstream, and this blimp, okay, this change gets restored. Okay, so so that's why the river, if you have a long river, and if you just have a few people live next to the river, and the sewage gets in, yeah, the river can purify, and then because the microbes in there, okay, the water then is still clean. However, if you have too many people dumping sewage into it, then this river becomes this. Okay, so if you go to upstate and you see a pond with all these growth, it's typically associated with too many people, too many houses next to the yard, next to the pond. And if you, so that's fresh water. The seawater isn't that. It's also have microbes. So if you take a boat out into the middle ocean, and the water is crystal clear, and you think it's clean, sterile, but no, they have bacteria, and they have archaea, and this is the numbers of bacteria. It's like 100,000 ml, 100,000 bacteria per ml. And this is archaea, but it's less. And you can, in the open ocean, what thrives are these nitrogen-fixing cyanobacteria, because they fix nitrogen, they don't need nitrogen. Because the river carries a lot of, like Hudson River carries a lot of waste from land, from it's waste for us, but it's nutrient for the microbes. So if you look down from the earth, this is what you will see. Okay. Can you see where we are? this is looking at chlorophyll and where we are here. Look at how far you have to go into the ocean to get away from microbe. And this is the area with lots of microbe growth because high density people release lots of nutrient. So that's on the surface. But if you go deep into the ocean floor, you will still see lots of microbes. And that's because the bottom ocean provides the temperature and the nutrient as the energy source. Okay. And more specifically, you can see there are tube worms. If you look at the Discovery Channel, you will see lots of underground tube worms. And these tube worms actually depend on microbes inside to provide nutrient for them. Okay. It's somewhat similar to we depend on nutrient in our gut to provide us from a nutrient to us, right? So if you cut open these microbes, you can see bacteria inside their cells. Okay, so those are the environments that I wanted to talk to you about. The soil, the fresh, the marine, and surface, and the bottom. Now, once we understand these environments, can we do about it? What can we do about it? And one, I want to tell you about two examples. One is the industrial use, and then the other is the agriculture use. So industrial use is that, well, we all love copper, right? And these days, copper are everywhere. You need electricity, and so the copper becomes very, very important. However, where do we get copper? Well, copper comes from stone or mines, right? Ore. And so some of the coppers, rocks have so little copper in there, it's just not energy worth to get it, to grind it up and extract. So then what they do is they use microbes to get it. So if you look at this picture, this is actually a copper mining operation using microbes. Really, yes, this is a hillside, it's a mountain. And if you look close enough to here, if you zoom into the picture here at the bottom, okay, and this is what's coming up. At the bottom of the pile of that rock or the hill, you will see a person there, and you will see liquid comes out. And if you look further, and this is at the bottom of the hill, they have liquid like floating through. Those liquid will float here. And then at the end of that, you will see a black stuff. And this is the copper. Now, it's not really pure copper, but it's highly enriched copper, and that you can use to extract copper. So how does that actually work? And so what does the microbe do? And so what happened is if you start from that liquid, and what happened is you start here, you want to pipe in. So if you get the liquid and you want to have ferric acid, and you have ferrous, all right. So you have ferrous, and you want to oxidize it, and you put the acid. So you go from ferrous ion to ferric, and using these two bacteria. And these two bacteria can oxidize it. And then the ferric ion acid, then you add sulfuric acid. So now you have sulfuric acid and ferric ion, and then goes up here. And then you sprayed, what's happening is you spray the rock with all these copper ores, with all these, this solution that has ferric in that. And then you will happen is that these reaction will happen naturally. The copper sulfur will be oxidized, and you have this copper sulfur. And then when you spray this on here, and you see the copper gets oxidized, and then these oxidized copper comes down, flows in that greenish water, comes down here, and then you provide the actual metal. And then they have reactions, and so the ion from the steel can gets replaced by its reduction assay. Oxidative and reduction assay remind you to review your chemistry reaction. And so now the copper becomes the metal. The ion gets to become the ferrous ion. Okay, so that's that black pile. It's actually copper. And so for this to work, then the water will pump back into the pool, and that the ferrous gets oxidized again. So as long as you have a pipe and you provide some sulfuric acid, you can continue to run this operation non-stop. Okay, so if you run this operation non-stop, and so this pipes up and you spray up there, and then comes down. Okay, and so these are eventually all come down, and so this liquid contains copper ion, and then the copper ion liquid comes here. You actually literally put the can with metal can here, and then you have exchange, and then the copper comes out into the elemental form instead of from the liquid. And so you sweep together, now you have copper. So that's an industrial use. What about agriculture use? Well, earlier we mentioned a couple of times that the life depends on the nitrogen. So if you don't have the first most complex thing we talked about is fixing nitrogen fixing air from the air, right? The azotobacter that fix nitrogen in the air. And I said that's an energy demanding process, requires lots of carbon source. And so, so some bacteria and some plants found a way to solve that problem. And so what happened is, let me do the backwards. So some bacteria, some plants like soybean, all the bean plants, soybean, lima beans, all these plants that produce beans, they belong to one family called legume. And what they do is they form, and also the grass, the yellowish grass. So if you remember Lehman College, right, the grass, the soccer field, in summer you have these white flowers, right? These white flowers are formed by clover, and these clover actually forms nitrogen fixing symbiosis with a bacteria. So what happens is on the root, they have these pinkish nodules. And these nodules of inside these nodules, later on I'll explain that these nodules will have bacteria inside and fix nitrogen. And so if you look backwards, the question is, how do they actually form these nodules? And these nodules are actually formed because bacteria get in them. So this is a diagram showing you the root hair. All plants have root hairs. And so on the root hairs, and if you have bacteria, rhizobium, colonizing on there, and the bacteria will produce a molecule called knot factor. The knot factor will cause the bacteria, cause the plant root hair to form curl. And then inside these curved root hair, you will have, so now you have curled root hair, and these bacteria will induce the formation of infection threads. See infection threads? Okay. These are the infection threads. Then inside these infection threads, you can have bacteria. Okay, so you will have bacteria, bacteria, bacteria. I'm trying to label it green, and so these bacteria going here, 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 green. Okay, I'm labeling. So this is just telling you the bacteria actually inside these tubes, and eventually the bacteria reach to the middle of the root nodule. All right, so the nodules are is a molecule that produced by bacteria, and all the genes producing these nodules are clustered together. This is similar to the theme we always talked about. Bacteria tend to put all genes required for one particular process together, and that's an example. So the knot factor looks like this. Okay, it's a repeating units of carbon, and so this is the common carbon molecule, and except that the different bacteria have different modification groups. So you have two different bacteria. One is cyanobacteria, Cyanorhizobium malodi. This is the bacteria that we study, and this is the bacteria that colonized the white clover on the grass, lemon play, the baseball field. And then you have another bacteria, and this bacteria colonized soybean, legume serum. And these two bacteria actually are different. They actually, the nitrogen fixing symbiosis requires a strict pair between bacteria and plant. They're very close, just like you have your best friend. Okay. And so what's unique about this is that the nodulation factors actually have different modification group to allow the plant to recognize bacteria. So white clover on Lehman College will only recognize Cyanorhizobium malodi, but not this bacteria. The reason is that the knot factor is the same, but the modification group is different. Just like you can recognize your friend by voice. If someone calls you on the phone, you don't recognize the phone number, and you pick up, you can tell who's your friend, right? Because we each speak differently. Our voice has unique signatures. So the signature of bacteria is embedded in these knot factor. What about plants? Well, plants also produce different molecules. And so this is the molecule, the green are the molecule that induces knot factor. And the red are the ones that suppresses, okay, don't induce. So when bacteria detect these molecules. Then they say, oh, my friend is here. Let me produce factor. And so it's just like you and your friends start talking. The more talk and the more you get excited, right? Normally what happens is if you start a small talk and you talk more excitedly and your voice gets louder and louder, that's similar to this, okay? And so the bacteria and plant exchange signal, and then they recognize each other, and then they form symbiosis. So let me summarize what actually happened. So this is a cartoon tells you, so this is alfalfa or white clover on the Lehman baseball field. And so the bacteria is there, and the plant produces flavonoids. Bacteria senses flavonoids and responded by producing nod factor. So you talk and I talk, so we recognize each other, and then the entering starts. So it's like your friend comes through your door, they buzz, and you talk on the intercom and say, ah, yes, my friend, let me buzz you in. You open the door, that's you open the door. And you let them go into your stairwell and go up. In this case, the plant actually literally builds a tube so they can come in. Okay, the tube is actually built by plant. The bacteria just grow inside the plant, the tube. And this, the infection thread and the bacteria, and so then the infection thread keeps going, and at the same time, the plant is growing and building a construct called nodule from nowhere. So the plant actually built this structure because there's a bacteria there. And inside these structures, some of the plant cells are colonized with a layer of bacteria. Okay, so here now you understand this is a root hair, and these are the bacteria that's growing into the plant, and that will lead to the formation of nodule. And then inside these nodules, and these are the plant cells, this is individual plant cells. Okay, you see individual plant cells. And these are the bacteria that turns into bacteroids, specialized. And you can see this bacteroids, this plant doesn't, this nodule have no bacteria. And so this fixed nitrogen and this does not. And so if you go in here, you can actually see it. This is a picture that we made, our lab made, many years ago using an SEM that we have in the department. And so what's actually happened is this. So you may get dizzy about this this picture, but as it turned out, you know majority of the stuff. So if we start from the beginning, and what's happening is this is the bacteroids. Okay, so if we were to focus on this one particular structure, bacteroids, what's in here? All right. Now, what's inside? And so if you look at this bacteroids, okay, so notice this bacteroids. Now the bacteroids, if we were to only look at the inside without going in, and the reason they're there is because they are producing this. Ammonia group comes out to plants. Okay? So this bacteroids gives plant the nitrogen, fixed nitrogen. Then what does this bacteria get? Well, fixing nitrogen requires energy, right? So this organic acid or sugar. Sugar with sugar, glucose, right? And that's the exchange. So the bacteria, so the plant gives the bacteria energy source. The energy source then being used by the bacteria to fix nitrogen and give it to the plant. All right. Now, in order for this process to go, it's more complicated than that. And if you see what's inside, now you're going to recall everything you've learned throughout the semester. Okay, so you have succinate, myelinate, and all these organic compounds. And then they go into the TCA cycle. And the TCA cycle cycles and comes out of the electron. Electron is carried out by the electron carriers, right? The NADH, the FDAH, and that goes through the electron transport chain, right? And creates, and then it creates the radiant, and then it generates the ATP. And the electron is ultimately dumped to the oxygen, right? And now in the process, it generates ATP, and the ATP then is being used by the nitrogenase to turn atmosphere nitrogen into ammonia. Okay? And this is, so now you actually know every single component of this complex diagram. It looks complex, but now if you calm down and look at it and say, aha, I actually know everything about this, right? Because this Bacteroids is a bacteria. It's a specialized bacteria. Okay, so I'm going to stop here. And just to quickly summarize, and these are the subject we talked about. We spent a lot of time talking about culture-dependent method of isolating bacteria, because that's somewhat difficult to understand. And so I spent a lot of time talking about that. And then we spent time talking about different methods, physical methods, and tracking bacteria. Then we talked about different environment, soil, freshwater, marine, and surface, and the bottom, the ocean. And then we talked about applications, the industrial application and the agricultural application. Okay, so I'm going to stop here, and I'm going to stop the recording.